Hello friends, welcome to my course on microbial biotechnology. Today we are starting module number 11, which will deal with the role of microbes in producing alternative energy. So in lecture 1, we are going to discuss about the production of biofuels using microbes. This lecture is divided into three broad sections. In section 1, we will be starting with an introduction to the topic. We discuss what is the need for alternative energy sources, how we utilize biomass, then microbial biofuel production from various feedstock, then technologies available for converting biomass into energy, definition of biofuels, types of biofuels, microbes involved in biofuel production, role of microbes in biofuel production, pretreatment of solid waste, why microbial pretreatment is important, then the various fungal enzymes available for lignocellulose degradation and lignin degradation by ligninolytic enzyme systems, hemicellulose degradation by hemicellulitic enzyme systems, and cellulose degradation by cellulitic enzyme systems. We will describe the remaining two sections as and when we will be coming across them. So, a significant portion of the energy utilized to support our daily lives is derived from non-renewable sources like coal, natural gas and petroleum. These resources, while currently meeting our energy needs, pose multiple challenges because they are finite. Lands are depleting faster due to population growth and infrastructure development, and their utilization contributes to environmental pollution by releasing harmful substances into the air, water and soil. So, here we can see the carbon dioxide emission from various power plants compared to geothermal energy, natural gas, oil and coal. Coal is the most polluting one, followed by natural oil and natural gas. The entire life cycle of these fossil fuels encompass mining, transportation, refining and consumption, which leaves behind a substantial carbon dioxide footprint, thereby exacerbating the global warming phenomena. Addressing these issues is imperative for fostering a sustainable and healthy future. A key solution is replacing fossil fuels with clean, renewable energy sources. An alternative approach involves the utilization of biomass, enabling the production of biofuels by microbes. These biofuels can match or even surpass the performance of fossil fuels. Let us examine the need for alternative energy. It is important because of environmental concerns. Reducing dependence on finite resources like fossil fuels and alleviating climate change is important. There is a global energy demand. Increasing human population and connectivity have led to higher energy demands, and current sources may not be sufficient. Diversification for sustainability is important. Exploration of alternative sources like sunlight, photovoltaic and thermosolar, wind, ocean tides and microbes are important. Then from the technological advancements point of views, advancements in technologies have made alternative energy sources more viable and efficient. Let us now focus about the utilization of biomass. Biomass is a total cellular dry weight or organic material an organism generates, usually from carbon dioxide and sunlight. Mostly these are plant based or algae based. Waste biomass is categorized into starch, lignocellulosic and oil fat biomass. Various microorganisms convert these type of organic biomass into biofuels like bioethanol, biodiesel, biogas, biohydrogen, butanol, acetone and advanced biofuels. Starch and lignocellulosic biomass are pre-treated with microbes or enzymes. To break down their structure, microbes directly convert water soluble sugars into biofuels, while oils and fats are trans-esterified to produce biodiesel. Let us look into the microbial biofuel production from various feedstock. It includes industry waste, then we have the biofuel crops, then we have agricultural waste and also the oil crops directly. So, waste from industries like paper and pulp generate a lot of kraft lignin and lignosulfonate, which can be converted into biofuels. Then we may directly grow biofuels like eucalyptus or switchgrass, then sugar and starch producing plants which can be converted into ethanol, which is actually the first generation liquid biofuel, while biofuel crops like eucalyptus and switchgrass provide us with second generation biofuels by providing cellulose for bioethanol production, or they may be used for pyrolysis to produce biochar and syngas like hydrogen, carbon monoxide and carbon dioxide. Then we have a large chunk of waste that is generated out of the agricultural produce. So, we term them commonly as agricultural waste, which may be solid biomass, lignocellulosic material, or it may be wet biomass, or it may be droop endocarp. All these, for example, the solid biomass gives us the scope for having the bioethanol from cellulosic material, and these are often referred to as the second generation biofuels. The wet biomass, particularly the organic waste manure, is used for production of biogas. Then the droop endocarp like olive, eastern black walnut, peach, coconut, etc., can be used for producing bioelectricity. Then we have oil crops which directly produce oil like rapeseed, corn, sunflower, soybean, coconut, palm, cotton. These vegetable oils can be directly converted into biodiesel. So, we can see that there are various feedstocks from which we can produce biofuel, but these processes involve the action of microbes. That is why we call them as microbial biofuels. Now, what are the technologies for converting the biomass that we just discussed in the earlier slide into energy? So, we have these diverse biomass resources which we need to do pre-treatment. Then we have the solid waste also, which may be directly used for thermochemical conversion by the process of pyrolysis and gasification. We can also use these biomass which is obtained by the treatment into thermochemical conversion, and by the process of pyrolysis we can get gas, oil and charcoal, and by the process of gasification we can gas the syngas. And we have these lignin cellulose and hemicellulose obtained by the pre-treatment, which can be used for biochemical conversion either by microbial fermentation or anaerobic digestion, producing ethanol, butanol and biogas respectively, and these all are used as power sources or fuel sources. In our lecture here we will be focusing mostly on these biochemical conversion part. So, let us have a small definition of biofuels. So, we can see that they can be so diverse depending on the biomass. Biofuels are fuels produced by biological agents from biomass, which could be solid, liquid or gaseous, primarily used in transportation as an alternative to non-renewable fossil fuels derived from petroleum. Biofuels offer a solution as fossil fuel supplies are declining rapidly. Economic and industrial growth in developing countries has increased fossil fuel consumption, further highlighting the need to consider biofuels as a sustainable replacement. So, we can categorize biofuels into 4 different types based on the biomass used. They can be first generation biofuels like the carbohydrate rich crops, sugarcane for example, Saccharum officinarum, and then sugar beet, Beta vulgaris, GM maize or corn. Then we have the oil rich crops like Brassica nepus, then we have the peanut, Arachis hypogea, and then Cocos nucifera, coconut, and we may also use the oil palm. Then second generation biofuels are the non-combustible and waste biomass, corns, stover, agricultural waste, forestry and sawmill residue, waste plant and span cooking oil, then rendered animal fats are some of the examples of these second generation biofuels. Then we have the energy crops like Mycanthus sinensis, then we have Panicum virgatum, or we may have sweet sorghum or Jatropha carcass. Then we have advanced biofuels or the third generation biofuels like macroalgae, microalgae, cyanobacteria, oleaginous microbes, and the fourth generation biofuels which are the genetically engineered microbial cell factories. Microbial biofuels are renewable energy sources derived from microorganisms. Bioethanol is produced by Saccharomyces cerevisiae fermentation and is widely used as a gasoline additive to enhance combustion and reduce emissions, and butanol is synthesized by Clostridium bacteria and offers higher energy content and better engine compatibility than ethanol. Biodiesel is made by transesterifying triacylglycerides from oils or fats into fatty acids, methyl esters or FAMEs by some oleaginous microbes. The microbes involved in biofuel production can be bacteria like Clostridium and Gymnosus mobilis, which produce various biofuels, or it can be algae, which are photosynthetic microorganisms that convert sunlight into biomass and produce lipids suitable for biodiesel production. Then we have yeast like Saccharomyces cerevisiae, which is used in the process of fermentation to produce bioethanol from sugars. Then we may have engineered microbes like E. coli and yeast, which are engineered to produce hydrocarbons and advanced biofuels. What is the role of microbes in biofuel production? Microorganisms used in several steps of biofuel production. For example, we may have solid biomass, which may be further divided into fats and oils, lignocellulose, sludge, starch and sugar. So, these fats and oils can be transesterified. These lignocellulose, microbial hemicellulosic biomass hydrolysis is done. Similarly, starch also hydrolysis is done. So, these undergo microbe pretreatment, and then we may use them for the cultivation of algae seaweeds, from which we extract the lipid, and that can be further transesterified. And then some part of these pretreated lignocellulose, sludge and starch can also be directly fermented by both aerobic and anaerobic, which give rise to the biofuel, biogas and liquid biofuel. So, we may obtain sugar directly, and that also can be fermented and produce biofuels. So, the pretreatment of solid waste by microbes are very important, particularly solid waste from agricultural, municipal and forestry sources often contain hemicellulose, lignin and cellulose, which are difficult to biodegrade. Pretreatment is required to prepare this biomass for fermentation. Biological pretreatment with microorganisms, especially white rot fungi like Phanerochite chrysosporium, offers a sustainable, cost effective alternative to chemical methods. Microbial pretreatment is preferred because it is sustainable, it is eco-friendly, rely on natural processes without producing harmful byproducts or hazardous waste. So, there is reduced chemical use or minimal use of synthetic chemicals that have less environmental impact and also reduces the cost involved. Then it also utilizes lower energy. So, because these microbial processes operate at ambient conditions, therefore, it requires less energy than thermal or chemical methods. These are cost effective methods. It do not involve costly chemical reagents, and the energy expenses are also cut down, making the process much more economical. And then there is improved biodegradability due to microbes, which selectively degrade lignin and hemicellulose, increasing cellulose availability for efficient biofuel production. There are fewer inhibitory substances when microbial pretreatment is done. It simplifies the fermentation process and reduces cost compared to chemical methods. Let us now discuss about fungal enzyme technology for lignocellulose degradation. Fungi have several biotechnological potential across diverse industries. Their effectiveness in degrading raw materials containing lignocellulose is attributed to their high efficient enzymatic systems. In addition to hydrolytic enzymes such as celluloses and hemicelluloses, which play a role in the degradation of polysaccharides, fungi possess a distinctive non-enzymatic oxidative system. So, here you can see the illustrative representation of cellulose, semicellulose and lignin, cellulose mostly having the glucose units, and when it is broken down we can obtain the glucose. Then hemicellulose contains xylose, arabinose, which are pentose sugars. Then it has also the hexose sugars, mannose, galactose and glucose, and they may be arranged in different permutation combinations. And then we have the lignin, which is having coniferyl alcohol, then para-coumaryl alcohol and synapyl alcohol as its constituting units. These in conjunction with lignolytic enzymes is responsible for modifying and degrading lignin. Lignin degradation by lignolytic enzyme systems is one of the most important step in this process. This is primarily an oxidative process which is often linked to secondary metabolism or limited carbon, nitrogen, sulfur available conditions. It is rarely utilized as a sole carbon and energy source. Basidiomycete white rot fungi are the primary agents for lignin degradation in nature. These fungi produce key lignolytic enzymes like lactases, then manganese peroxidases, lignin peroxidases and versatile peroxidases. So, these lactases are blue multi-copper enzymes with the ability to oxidize a diverse range of phenolic and non-phenolic compounds. These illustration depict the orientation regarding copper atoms and the ligand distance. The blue distances in Armstrongs, the 3-dimensional structure used for lactases in Coriolis jonatus, which are obtained from PDB. They catalyzed the 1 electron transfer of phenolic compounds including lignin, polyphenols, methoxy substituted phenols and diamines, forming radicals that can either repolymerize or undergo depolymerization. They exist as monomeric, dimeric or tetrameric glycoproteins, typically containing 4 copper atoms per monomer distributed across 3 redox heights named T1, 2 and 3. In their resting state, all 4 copper ions are in the +2 oxidation state, as you can see in the figure. The majority of lactases are produced by white rot fungi secreted into the medium by the mycelium of filamentous fungi. Some examples of fungi known for producing lactases with high activity includes Tremates pubescens, Coriolis hirsutus and others. So, here you can see the lactase catalytic cycle. Substrates are oxidized by the CuT1 center and electrons are transferred by a highly conserved motif his cis his to the T2 and T3 copper centers. This is where the reduction of molecular oxygen to water takes place. Let us now discuss about the hemicellulose degradation by hemicellulolytic enzymes. The hydrolysis of hemicellulose requires the coordinated action of multiple enzymes that target various components within the hemicellulose matrix. So, here we have different enzymes with their EC number and the substrates corresponding to these enzymes and the various functions listed in these tables. For example, beta mannosidases, which has the EC number of 3.2.1.25. The substrate for these mannosidases is mannose containing oligosaccharides. They hydrolyze beta D mannosidic linkages, breaking down mannans and mannan containing oligosaccharides. Then we have the beta xylosidases, which hydrolyze beta D xylosidic bonds in xylan or xylo oligosaccharides, thereby releasing xylose. Then we have the beta mannanases, which hydrolyze beta D mannan into smaller oligosaccharides or monosaccharides. Then we have the beta xylanase, which cleaves internal beta 1,4 glycosidic bonds within the xylan chain, producing shorter oligosaccharides. So, here this figure shows enzymatic hydrolysis involved in the breakdown of cellulose and hemicellulose biomass. So, here we have these hemicellulose where the hydrolysis begins with an endoglucanases attack, as you can see here. Then these oligomers are available for the exoglucanase action, where this is cleaving this. Then we have this crystalline region where the cellobios are released. So, these basically the beta glucosidases will finally release the glucose. Then here again we can see the action of endogyluconases taking place that releases the xylobiose, and beta glycosidases will thereby release the xylose, and the action of beta mannanases will give rise to manno-oligomers which will be acting upon beta mannosidases to release the mannose. So, this is in brief the enzymatic hydrolysis in the breaking down of cellulose and hemicellulose biomass. Fungal degradation of cellulose relies on a suite of glycoside hydrolase enzymes, each with distinct catalytic roles. Cellulase possesses a carbohydrate binding module that facilitates binding to crystalline cellulose. Based on their mode of action and specificity, cellulases are categorized into three primary classes: endoglucanases, exoglucanases, and beta glucosidases, having distinct EC numbers, and they act on cellulose beta 1,4-glucan and cellobiose beta 1,4-glucosidic bond, and the functions are listed corresponding to endoglucanases. It hydrolyzes internal beta 1,4-glycosidic bonds in cellulose to produce smaller oligosaccharides. Exoglucanases cleave cellobiose from the non-reducing or reducing ends of cellulose chains. Beta glucosidases hydrolyze the disaccharide cellobiose into two glucose molecules. Let us now go to section 2, and here we will discuss about some of the major biofuels and how bioethanol is produced, how biodiesel is produced, and how biogas and biohydrogen are produced. So, as we can see, the major biofuels include bioethanol, biodiesel, biogas, and hydrogen gas. Bioethanol is produced by microorganisms from biomasses like starch or sugar crops through enzymatic and chemical hydrolysis, and has drawn scientific attention as a potential gasoline replacement or additive. Through though currently not cost competitive with petrol, it is used for transport due to governmental subsidies and its sustainable nature. So, for production of bioethanol, we have to have starch feedstock which is hydrolyzed, that process is also known as liquefaction, and then we go for the saccharification step, whereby we obtain the fermentable sugars that is acted upon by the yeast to produce bioethanol. So, Saccharomyces cerevisiae is preferred for bioethanol production due to its tolerance to high ethanol concentration and inhibitors. Bacteria like Escherichia coli and Gymnosus mobilis are other commonly used microbes for ethanol production. Saccharomyces cerevisiae and Gymnosus mobilis are commonly used microbes, but neither can ferment xylose, a major sugar in lignocellulosic biomass, hydrolyze it. Efforts to genetically engineer Saccharomyces cerevisiae to ferment xylose have shown promise, with genes from Pichia stipitis enabling xylose conversion to xylulose. However, no engineered strain has reached commercial production till now. So, the microorganisms exhibit distinct metabolic pathways and utilize various catalytic enzymes to generate biofuels. The universal pathway for sugar breakdown, basically the glycolysis and fermentation, where glucose is converted to pyruvate and then acetaldehyde, then finally to ethanol with the help of yeast and bacteria. As glycolysis cannot process pentose sugars, pentose phosphate pathway comes into play. The xylose arabinose are converted to ribulose 5-phosphate, which are converted to many intermediates, and finally those are converted to pyruvate and finally converted to ethanol. And in this case, Pichia stipitis and engineered E. coli has been used. So, some of the pathways for the production of acetaldehyde pyruvate, which is an intermediate in the central metabolism of carbohydrates, can undergo conversion to acetaldehyde, a precursor ultimately reduced to ethanol through two distinct pathways. One is a two-step pathway. In this process, pyruvate undergoes non-oxidative decarboxylation to form acetaldehyde and carbon dioxide, catalyzed by pyruvate decarboxylase. Then subsequently acetaldehyde is transformed into ethanol through the action of alcohol dehydrogenase. Then there is a 3-step pathway which is more prevalent in bacteria. This pathway involves the oxidative decarboxylation of pyruvate to acetate coenzyme A by the metalloenzyme pyruvate ferrodoxin oxidoreductase and or pyruvate formate lyase. Acetyl coenzyme is then converted to acetaldehyde by a coenzyme dependent acetylating acetaldehyde dehydrogenase. Finally, acetaldehyde is reduced to ethanol by alcohol dehydrogenase. Let us now have a discussion on the next important biofuel, the biodiesel. Biodiesel is a diesel-like liquid from biological materials, typically made of fatty acid alkyl esters. It is an environmentally friendly alternative to diesel fuel, attracting attention for diesel engines. While alkaline catalysis is common for commercial production, enzymatic transesterification offers benefits like fewer processing steps and easier glycerol separation, though high enzymes costs are a limiting factor. Immobilizing fungal mycelium and expressing lipase on yeast cells helps reduce the enzyme cost to a large extent. So, the production process involves the transesterification of oil, and sometimes we also start with methanol, ethanol solvent, then immobilized lipase enzymes or potassium hydroxide, which is a chemical. These are all mixed and used in the transesterification process. Then we go for the phase separation, and the byproduct glycerol is separated out. By this phase separation, we get the crude biodiesel. We go for washing of these crude biodiesel and then drying, and finally obtain the biodiesel. So, the feedstock used in biodiesel production, particularly microbial feedstock, involve bacteria, yeast, fungi and microalgae with high lipid content, and oleaginous bacteria being less used compared to microalgae and yeasts. Among bacteria, Rhodococcus species is well studied for its versatile substrate growth and lignin degradation potential. Using algae for biodiesel is promising due to their easy cultivation, diverse metabolic activities and high fatty acid content. Algal species like Dunaliella, Tartiolecta and Chlorella are identified as potential feedstocks. Yeast strains like Candida lipolytica show high lipid content in certain conditions, and they are also potentially very good feedstock. The transesterification is the most important step in this whole process. The production of biodiesel from algae involves obtaining its lipid fraction by either mechanical approaches such as microwave assisted pyrolysis extraction, ultrasonic assisted extraction, or chemical methods involving organic solvents like chloroform, petroleum ether, acetone, methanol and hexane, as well as techniques like Soxhlet and supercritical fluid extractions. Following the extraction of algal lipids, the next crucial step is the transesterification of these lipids into biodiesel. Lipases facilitate the reverse reaction, transesterification, where 1 mole triglycerides reacts with 3 moles of alcohol, methanol or ethanol, to form biodiesel, fatty acid methyl esters and glycerol as a byproduct, as you can see in this reaction. Factors such as temperature, stirring rate, feedstock purity and reaction time contribute to improving biodiesel yield. Sometimes we will use immobilized lipases in this catalytic reaction. Using immobilized lipases in biodiesel production is a significant area of research and industrial application. Immobilizing offers numerous benefits that make them suitable for industrial scale biodiesel production. The first utility is that we can reuse the enzyme. The immobilization increases the stability of the enzyme. There is ease of separation of the enzyme, the catalyst, from the product. Immobilized enzymes show higher activity and we can use it for continuous operation. So, there are various methods of immobilization due to limitation of scope and time. We will not go into details of those. So, some of these can be binding them to carriers by simple adsorption or covalent binding, or ionic binding or by affinity binding methods. Another method is to entrap them into a certain matrix or encapsulate them into small particles which may be microparticles. Then also cross-linking the lipases with one another. Let us now discuss about the production of biogas, particularly anaerobic digestion. So, here we have these digestate which we will be using for the production of the biogas. Particularly we have agricultural waste which we use for hydrolysis reaction that produces soluble organic substances like sugar, fatty acid and amino acids which are used for carrying out acidogenesis to produce hydrogen, carbon dioxide and volatile fatty acids which are further subjected to acetogenesis reactions, and these products of these acetogenesis reactions are further treated for carrying out methanogenesis which produces the biogas. So, this is in brief process of biogas production by anaerobic digestion, which is a complex biological process involving interconnected biochemical reactions facilitated by diverse microbial communities. The key stages include hydrolysis, acidogenesis, acetogenesis and methanogenesis as already shown in the diagram. Hydrolysis is the initial step that involves the breakdown of complex organic compounds like carbohydrate, protein and lipids into simpler soluble compounds such as simple sugars, amino acids and fatty acids, followed by acidogenesis where these soluble products are further transformed into short chain fatty acids commonly called volatile fatty acids which typically have 2 to 6 carbon atoms. This phase is crucial as it lays the groundwork for subsequent reactions. Then the third is the acetogenesis where these volatile fatty acids produced undergo further anaerobic oxidation resulting in the production of acetic acid along with the release of carbon dioxide and hydrogen gas. These steps serves as an intermediary stage before the final conversion of these compounds into methane. In the final methanogenesis, we have the conversion of the produced gases particularly acetate and a combination of hydrogen and carbon dioxide into methane. This conversion occurs through two primary pathways. One is the acetoclastic methanogenesis where acetate is directly transformed into methane and carbon dioxide. Then there is the hydrogenotrophic methanogenesis. Hydrogen in combination with carbon dioxide is utilized to produce methane. The culmination of these various steps results in the production of biogas primarily composed of methane and carbon dioxide which can be harnessed as a renewable energy source. Some of the microbes that are used in anaerobic digestion are hydrolytic bacteria for the hydrolysis stage which includes Clostridium, Bacteroides, Bacillus actinomyces. Then the acidogenic bacteria which are involved in the acidogenesis like Escherichia coli, Clostridium, Lactobacillus and Streptococcus. Then for the acetogenesis stage we have the acetogenic bacteria like Syntrophomonas, then Syntrophobacter and Clostridium, and the methanogenic archaea carries out the methanogenetic reactions. So, these are mostly the Methanobacterium, Methanospirillum, then Methanosarcina and Methanoseta. Now, let us discuss about the production of biohydrogen which is produced by bacteria and algae and is mainly generated through anaerobic fermentation and photolysis of water. Photosynthetic microbes such as cyanobacteria, photosynthetic bacteria and green algae as well as non-photosynthetic bacteria like anaerobic and nitrogen fixing bacteria are key producers of hydrogen. Both non-photosynthetic and halophilic photosynthetic bacteria can produce hydrogen. The co-culture of Rhodiobium marinum and Vibrio fluvialis produces hydrogen comparable to the bacterial community, highlighting the significance of these bacteria in hydrogen production from stars. So, the production pathways for biohydrogen involve dark fermentation where certain bacteria like Clostridium breaks down organic matter like sugars or starches anaerobically to produce hydrogen gas. In this process, organic acids such as acetic acid and butyric acid are produced alongside hydrogen, which can be recovered after fermentation. Then in photofermentation, photosynthetic bacteria like Rhodobacter species produce hydrogen in the presence of light using organic substrates like organic acids. During photofermentation, light energy drives the production of hydrogen through the light dependent reaction. Then there is biophotolysis, where microalgae or higher plants utilize sunlight to split water molecules into hydrogen and oxygen in a process similar to photosynthesis. This method is still under research as it requires efficient methods to optimize and harvest hydrogen from algae. Then we have microbial electrolysis cells, which we will be discussing in the next lecture. Briefly, in MEC, bacteria used to reduce protons in water and generate hydrogen gas. This is an electrochemical process where bacteria interact with electrodes in a cell that applies a small electrical current, facilitating the production of biohydrogen. Let us now go to section 3, where we will be discussing briefly about metabolic engineering for enhancing biofuel production, then genetic engineering of lipid metabolism in microalgae, then sustainability and environmental benefits of biofuels, and challenges in microbial biofuel production. Metabolic engineering involves modifying the metabolic pathways of microorganisms to enhance biofuel production by improving yield, substrate utilization and process efficiency. Bacteria like Zymomonas mobilis is efficient in bioethanol production via the entero-Dodorov pathway, but struggles with pentose sugar utilization. Genetic modifications have improved its ability to utilize lignocellulosic biomass. Another bacteria, E. coli, was engineered for ethanol production by incorporating genes from Z. mobilis and disrupting competing pathways, however, requires complex nutrients and is vulnerable to phage infections. Overexpressing acetyl coenzyme A carboxylase in E. coli increased free fatty acid production by 20 times, which enhanced biodiesel production. Then there are attempts at genetic engineering of Saccharomyces cerevisiae to enable fermentation of more complex sugars that usually require pre-treatments, thus making bioethanol production more efficient. There are also attempts at genetic engineering of lipid metabolism in microalgae. Enhancing lipid production by overexpression of certain genes has been shown to increase lipid content in both plants and microalgae. For instance, overexpressing G3 phosphate dehydrogenase in Brassica napus resulted in a 40% increase in lipid production. Then blocking competing pathways is another approach in microalgae. Blocking starch production pathways, example in Chlamydomonas reinhardti, can divert resources towards triacyl glycerol accumulation, especially under stress conditions like nitrogen deprivation, thus increasing lipid content. Then modifying lipid catabolism is also one strategy. Reducing lipid catabolism by inactivating genes involved in fatty acid degradation can increase lipid accumulation. However, this often results in growth inhibition, as the breakdown of lipid is also crucial for energy production. Modifying lipid characteristics by genetic modification, which are aimed at changing the fatty acid profile, such as shifting chain lengths or unsaturation, can enhance biofuel suitability. Overexpression of thioesterase races from Umbularia californica in microalgae can produce shorter chain fatty acids ideal for biodiesel, while others aim to produce short chain fatty acids for gasoline or jet fuel production. Biofuels benefits us in many ways, particularly from the sustainability and environmental point of view. Biofuels helps us in reduce carbon emission. It can significantly decrease the emission of carbon dioxide, thereby mitigating climate change impacts. It helps us in carbon sequestration, particularly use of plant biomass for biofuels can help in sequestrating carbon from the atmosphere. Then it can help us by offering us carbon credit trading. By producing biofuels, countries like India can earn carbon credits, especially if they avoid emitting carbon dioxide. Then there will be reduced vehicular emissions. Ethanol blends in gasoline to reduce tailpipe emissions including carbon monoxide. However, there are many challenges in microbial biofuel production. It suffers from low yield and productivity. Limited biofuel production rates in engineered microbes make it economically unfeasible at large scales. the cost of raw materials, high cost of feedstocks like lignocellulosic biomass and waste oils due to complex processing requirements. Then there are the technical challenges where genetic stability, particularly engineered strains, may lose biofuel production traits over time, and challenges like scaling up, difficult in translating lab scale processes to large industrial systems. And then the environmental concerns, competing land use, in particular biofuel crops may compete with food production, raising sustainability issues. And then the sustainability of feedstocks, potential environmental impacts from large scale feedstock production, example water use, soil depletion, etc. So, with this we come to the end of this lecture. Thank you for your kind attention.